So my name is Dena. I'm a third-year student in the Libo lab, and I'm studying the role of microglial D-Serine in synaptic damage following traumatic brain injury. So just to start with a little bit of background information on TBI. TBI occurs when there is damage to the brain, and this can be either due to a direct mechanical impact, rapid acceleration and deceleration of, as well as shock waves, which are most common in military settings. And TBI affect almost 70 million people worldwide each year, and they most commonly affect young children as well as elderly populations. And since there is such a wide array of mechanisms for obtaining a TBI, there is also a wide range of symptoms, such as cognitive impairments, which can be impairments to learning and memory, motor deficits, and behavioral symptoms, such as changes in aggression and greater risk for anxiety or depression. So as I said, there are many diverse mechanisms of injury after a TBI, and this is typically split into two phases, the primary injury phase, as well as the secondary injury phase. So the primary injury occurs at the time of impact, and this includes things such as direct tissue damage and damage to blood vessels and the blood-brain barrier. And since primary injury occurs directly at the time of impact, there is little that can be done to treat this injury. So when we talk about therapeutically targeting TBI, we're mostly referring to secondary injury mechanisms. And secondary injury occurs in the seconds, minutes, days, and even years after the initial impact. And this includes aspects such as ischemia and edema, excitotoxicity due to excessive glutamate release, which can lead to mitochondrial damage and oxidative stress. And what I'm going to be focusing on mostly in my project is synaptic damage and gliosis. So before I get into synaptic damage and gliosis in traumatic brain injury, just wanted to highlight the importance of glia and synapse interactions under normal physiological conditions. So under normal healthy conditions, astrocytes and microglia are constantly in contact with neurons and contribute to synaptic activity. For example, one of the most well-studied aspects of this is the tripartite synapse, where astrocytic end feet make contact with both the pre- and postsynaptic elements in the synapse and are very important for buffering glutamate as well as potassium and other ions to prevent excitotoxicity and control neuronal excitability. And astrocytes can also release gliotransmitters in response to synaptic activity to facilitate transmission and plasticity. And the role of microglia under physiological conditions is also being increasingly appreciated, where microglia are very important for synaptic pruning during development. So they're essential to proper refinement of neuronal circuits, and abnormal synaptic pruning by microglia has been implicated in a lot of neurodegenerative disorders, including schizophrenia, Alzheimer's disease, and autism spectrum disorders. And in the adult brain, microglia also constantly make contact with the pre- and postsynaptic elements of neurons, and this has been shown to occur in an activity-dependent manner, where microglia have recently been implicated in regulating neuronal synchronization. And microglia can also release gliotransmitters to contribute to synaptic plasticity, such as, for example, the transmitter BDNF. So in my project, I'll be focusing on synaptic plasticity, in particular, NMDA receptor dependent synaptic plasticity. So NMDA receptors are expressed postsynaptically, and they can bind glutamate that is released from the presynaptic terminal during normal synaptic transmission. And these glutamate receptors are composed of two main subtypes, the GLU-N1 subtype, which is found in all NMDA receptors, as well as the GLU-N2, which has two different subtypes, which I'll get into a little bit later. And glutamate, these NMDA receptors are unique in that they, in addition to binding of the agonist glutamate, they require a modest depolarization as well as binding of a co-agonist, which in the hippocampus is D-Serine. And once all three of these conditions are met, this leads to influx of calcium into the cell, which can then lead to changes in long-term potentiation and gene expression. And in cases of injury, excessive calcium increase can lead to excitotoxicity. So for my project, I'm focusing on the role of D-Serine in synaptic plasticity after injury. And D-Serine is a D-amino acid, which functions as the co-agonist of NMDA receptors. And D-Serine is normally synthesized by neurons by the enzyme serine racemase, shown here in yellow. And serine racemase converts L-serine, a precursor which is synthesized in astrocytes and then shuttled into postsynaptic neurons. And it converts this L-serine into D-Serine, which is then released during normal synaptic activity and can bind NMDA receptors. And in addition to making the precursor L-serine, astrocytes also play a role in uptake of D-Serine as well as its degradation. So since D-Serine is so important for synaptic function under normal conditions, our lab is interested in its role following traumatic brain injury. So to look at this, we use a controlled cortical impact model, where a mouse is placed in a stereotactic frame and a highly pressurized piston delivers a mechanical impact over the intact dura. So as you can see here, in CCI conditions, there is severe loss of cortical tissue as compared to a sham brain. However, the hippocampus remains intact. And when we looked at the number of new and positive cells within the hippocampus, we don't see any significant changes between the sham mice in white and the CCI mice in black. However, when quantifying the number of synapses in the CA1 region of the hippocampus, there is a significant decrease following injury. So this model, therefore, is really advantageous to study synaptic damage and remodeling after injury in the absence of cell losses. And here, this is just showing the functional effects of the CCI injury, where we see a loss of long-term potentiation in the CCI mice in black as compared to the sham mice. And looking at this from a behavioral standpoint, we use the fear conditioning paradigm, which I'll talk about in more detail later. But essentially, it is a contextual fear conditioning paradigm where mice are placed in a context and their percent freezing is measured as an outcome of their ability to associate an aversive stimulus with either a context or cue. So in this case, more freezing indicates better learning and memory. So you can again see a deficit in our CCI mice in black, showing that these synaptic losses do have functional outcomes. So to begin to look into the role of D-Serine in these synaptic deficits after injury, we first looked at serine racemase expression in sham and CCI conditions. And as you can see in the top left, in sham conditions, serine racemase is expressed predominantly in neurons in this molecular layer versus 3 days after injury, it has a more diffuse profile. And when looking at staining with GFAP, which is an astrocytic marker, we see this increase in GFAP expression after injury, showing that there is increased astrogliosis. And we do see increased co-localization of serine racemase with GFAP positive cells after injury. So we had previously shown that these astrocytes upregulate serine racemase expression in our CCI model. And we have shown in a previous paper in 2017 that ablating serine racemase in astrocytes leads to improved functional outcomes after injury. So today, I'll be focusing on microglia as a novel source of serine racemase after injury, where similarly, we look at IBA1 expression 3 days after injury, we see this increased expression here in blue. And this is a microglial specific marker showing enhanced astrogliosis, microgliosis in our model. And when looking at the colocalization of serine racemase and microglia, we do see this colocalization in the 3 and 7 days after injury. And this was quantified using flow cytometry as well, where you see a significant increase in the percent of microglia that express serine racemase at 3 and 7 days after injury, as well as an increase in the mean fluorescent intensity after injury as well. So not only do more microglia cells express serine racemase, individual cells express more as well. So this is the first in vivo evidence that microglia cells can express serine racemase. So given this novel cellular source, we wanted to know whether this microglial D-Serine causes synaptic damage after injury. So to do this, we used 2 different microglial serine racemase knockout models, where serine racemase is conditionally knocked out under the TMEM119 promoter and the CX3-CR1 promoter. And then we also have serine racemase tamoxifen control mice. So these mice receive either a sham or a CCI injury, and then in the 5 to 7 days after injury, undergo behavioral testing using fear conditioning. And at 7 days, we harvest this tissue to look at Golgi analysis. So Golgi analysis is the silver-based stain, which allows us to visualize dendritic spines. And this allows us to not only quantify the number of spines, it also allows us to look at the morphology, where we're interested in expression of these mushroom-shaped spines, which correspond to mature synapses, as they have this large surface area and can receive more synaptic inputs. And we're also interested in long, thin, and thin spines, which are a hallmark of degenerating spines. So when looking at dendritic spine density in the CA1 region of the hippocampus, we see that in our control mice, there is a significant downregulation in the spine density at 7 days after injury. And we see similar effects on spine morphology, where we have a significant decrease in the percentage of these mature mushroom-shaped spines, and a significant increase in the percentage of thin spines. And when looking at this in the microglial knockout mice, we see that this effect is ablated, where there is no decrease in spine density after injury and no changes to spine morphology, suggesting that microglial serine racemase ablation can rescue dendritic spines. So to see whether this had a functional correlate, we used the fear conditioning assay, which at 5 days, the mice are habituated to this cage, and then on the 6th day after injury, mice are placed in the cage and they receive an auditory stimulus followed by a 3-second long foot shock. And then 24 hours later, they're placed back in the same apparatus. And we measure the percent freezing of the mouse, where freezing is considered to be a fear-based response. So an increased percentage of freezing in this case corresponds to a better ability to associate the context with the aversive stimulus. So in our control mice, we see significantly less freezing at 7 days after injury. And in both microglial knockout mice, this is once again ablated. So showing that ablation of microglial serine racemase can rescue functional outcomes as well. So to summarize everything that we know so far, in normal, sham, or healthy conditions, serine racemase is expressed postsynaptically, where it converts L-serine to D-serine, and D-serine is released to bind to NMDARS receptors for normal synaptic transmission. However, in a TBI, neurons will downregulate serine racemase expression, where both microglia and astrocytes upregulate serine racemase. And so we've seen that knocking out serine racemase in either microglia cells, as I just showed you, as well as in astrocytes, which the lab has previously found, is sufficient to rescue both dendritic spine densities, morphologies, and functional outcomes. And I haven't shown you this here, but we've also looked at ablation of these D-Serine transporters, specifically the SLC1A4 and SLC7A10 transporters, which are glial-specific transporters and are important and are a way for microglia and astrocytes to release D-Serine. So ablating either glial synthesis or release of D-Serine is sufficient to rescue synaptic function after injury. However, what still isn't known is what is the overall mechanism of this D-Serine mediated synaptic damage following a TBI. So this is what I'll be looking into for my project, where my overall hypothesis is that glial D-Serine mediates synaptic damage by hyperactivating extrasynaptic GluN2B containing NMDA receptors, and also can alter microglial responses following injury. So this can be broken down into two different hypotheses, where the first one is focusing on this hyperactivation of NMDA receptors. And the second part is focusing on the complement-mediated tagging of dendritic spines for removal. So focusing on the first part of my hypothesis, which is that astrocytes and microglia release D-Serine, which can then hyperactivate NMDA receptors, leading to the synaptic damage that we see. So just to go over a little bit about the different subtypes of NMDA receptors. As I mentioned before, NMDA receptors have two main subunits, the GluN1 subunit, which is expressed by all NMDA receptors, and then the GluN2 subunit, which there's two different versions that are mainly expressed. So one is the GluN2 version, which is enriched in synaptic NMDA receptors. And this has been implicated in pro-survival signaling as well as synaptic plasticity. So in particular, these synaptic GluN2A receptors facilitate upregulation of BDNF, which is important for plasticity, as well as ERK1/2 and Krebs phosphorylation, leading to transcription of these pro-survival genes. On the other hand, the GluN2B subunit is expressed predominantly in extrasynaptic NMDA receptors. And these receptors have low basal levels of activity, and excessive activation of these receptors in multiple disease models, such as ischemia, Alzheimer's disease, and Huntington's, has been shown to inhibit these pro-survival signals from synaptic NMDA receptors. And here, this is just showing that in our CCI model, we see an increase in this N2B subunit after injury. So to kind of begin looking into the role of the subunit, I started with pharmacological administration of Ro-maleate, which is a GluN2B specific inhibitor. So to do this, I use these osmotic mini pumps where you can fill them with your drug of interest. In my case, I use 3 different concentrations of Ro-maleate, as well as saline as a vehicle control. And then these pumps are implanted subcutaneously and connected to the lateral ventricle via a cannula, so that the drug is administered at a constant rate into the lateral ventricle. And so we administered these inhibitors for 3 days, at which point I harvested the tissue to look at dendritic spine analysis. And so here we can see in our sham conditions, these are just representative images of spines from the CA1 region of the hippocampus, where in sham conditions, there is a predominant expression of these mushroom-shaped spines, versus in the CCI condition, we see a much greater representation of these thin degenerating spines. And here, this is in a CCI mouse that was treated with the Ro inhibitor. We see ablation of these thin spines and more mushroom-shaped spines, similar to the sham condition. And when quantifying this, I did see a significant decrease in the percentage of mushroom-shaped spines in our vehicle CCI condition, which corresponded to a significant increase in thin spines. And this effect was ablated at all 3 concentrations of the Ro inhibitor, suggesting that this is a target for synaptic damage in our model. So right now I'm looking at whether this GluN2B specific inhibition using Ro-maleate can also rescue learning and memory at 7 days, as well as if this corresponds to synaptic preservation at this time point as well. I'll also be using a genetic knockout model where I'll be using GluN2B conditional knockout mice, where the GluN2B subunit is conditionally knocked out in CAMK2 positive neurons. And I'll be evaluating this similar to before using fair conditioning and Golgi, in addition to water maze and electrophysiology as secondary measures of learning and memory. And then finally, I'll be looking at whether glial D-Serine activates these GluN2B pathways after injury. So I'll be employing our TMM119 serine racemase knockout mice, where serine racemase is ablated specifically in microglia cells, and seeing whether there are differences in these signaling components, such as BDNF and phosphorylation of CREB and ERK after injury. So moving on to the second part of my hypothesis, which is that the hyperactivation of NMDARS receptors by glial D-Serine will lead to the complement-mediated tagging of dendritic spines for removal by microglia cells. So complement has been implicated in synaptic pruning in many cases in development as well as in disease, where this complement C1q, which is the initiator component of the complement cascade, can bind to synapses for removal. And C1q leads to recruitment of C3b, which functions as an opsonin. And together, these signals are recognized by receptors on microglia cells, leading to engulfment and elimination of the synapse. So just some preliminary studies that I did to look at this was looking at C1q expression in whole tissue before and after in sham and CCI conditions. So here in wild type mice, we see a significant increase in C1q expression after injury. And this effect is not present in the microglial knockout mice. And here we can see the same effect from an immunohistochemical standpoint, where you see this increase in C1q in green in the control mice after injury, which is attenuated in the knockout mice. So to go over this mechanism in a little more detail, we're hypothesizing that after injury, glial cells will upregulate serine racemase expression and thus release D-Serine to activate these extrasynaptic receptors, which normally have low levels of activity. So when they're hyperactivated, this leads to decreases in genes involved in survival signaling and synaptic plasticity. And one other component that I'm interested in looking at is phosphatidylserine. And phosphatidylserine is a phospholipid that is normally localized to the inner leaflet of the plasma membrane. However, under cases of cell stress, it is externalized on the cell surface, where it can then be recognized for phagocytosis. So this mechanism where phosphatidylserine is externalized specifically in synapses or in spines has been shown during development as well as disease. And C1q has been implicated to be able to bind phosphatidylserine. And together, these signals work through a novel receptor on microglial cells called GPR56. So it's been seen that this GPR56 receptor can bind phosphatidylserine, leading to synaptic removal. So I'll be looking into this as a potential mechanism of synaptic losses in my project. Where first I'll be looking at if D-Serine can alter the expression of complement components, as well as PS-mediated tagging of spines for removal after injury. So I'll be using our microglia knockout mice again to look at whether there are changes in these components in our knockout model as compared to controls. And I'll also be looking into the role of this GPR56 receptor in injury, where we're currently producing GPR56 knockdown mice to look at whether there is reduced synaptic losses after injury in these mice. And in addition to looking at this from a focus mechanism, I'll also be doing some experiments to look at the role of microglial D-Serine on a more global level, where first I'll be examining whether D-Serine has an effect on overall inflammatory responses. So previous studies done in cell culture have shown that exposure of microglial cells to inflammatory stimuli such as LPS and beta amyloid can induce serine racemase expression. However, the opposite has not been studied. So I'm interested in seeing whether knocking out serine racemase in microglia will lead to reduced overall inflammation after injury. And finally, I'll be looking at transcriptomic changes after injury, where I'll be conducting single cell sequencing on all the various cell types in the hippocampus in both control mice in sham and injured conditions and our serine racemase microglial knockout mice to see whether microglial D-Serine has downstream signaling effects on specific cell populations and subtypes of populations. So with all of that being said, everything I've shown you so far has been data from male mice. So something that I'm interested in looking at in my project is if there are sex differences in synaptic damage and if microglial D-Serine plays the same role in female mice as we've seen it plays in male mice. And I think this is a very important aspect to look at since a lot of studies have seen differential outcomes following injury in male and female mice. So to begin looking into this, I started with fear conditioning experiments, where in our serine racemase knockout mice, I did not see, or sorry, in our serine racemase control mice, we don't see a deficit in freezing like we normally would in male mice. And with our knockout mice, we also don't see any significant changes in freezing behavior. So when looking at Golgi analysis to look at spine density, also did not see any differences between the sham and CCI control conditions, as well as the knockout mice. And this is done at 7 days after injury, which is a time point where we usually do see losses in spines in male mice. When looking at spine morphology, there does seem to be a trend towards decreased mushroom-shaped spines, as well as an increase in the thin-shaped spines in CCI control mice. And this effect appears to be slightly attenuated in our knockout mice. However, it's hard to really determine right now with such a small sample size. But when looking at just control mice, looking at males versus females, we see that there is greater synaptic damage in male mice. So here in red, you can see this significant decrease in the mushroom-shaped spines in male mice, as well as a significant increase in thin spines after injury. And there is less damage in the female mice, suggesting that there is more neuroprotection in female mice after injury. And so I'm interested in looking into this further and seeing if there are differences in expression of serine racemase in microglial cells in female mice, and looking at levels of D-Serine in the whole hippocampus after injury in females to see whether this secretion of D-Serine plays any role in the neuroprotection that we see. And so in addition to looking at sex differences, another part of my project is looking into whether some of these aspects that we've seen in our CCI model are present in human TBI patients as well. So to do this, we've been collaborating with Dr. Grazioli and the neurosurgery team here at the University of Miami, Jackson Health System, where they have been providing us with tissues that are resected during neurotrauma surgery. And these tissues are usually from the perilesional space, and they are typically from cortical samples. So kind of the first thing that we wanted to look into is whether there are changes in serine racemase in glial cells in the mouse perilesional cortex, since everything we've looked at so far has been focused on the hippocampus. So here, when we just looked at immunohistochemical staining in the perilesional space, we see this increased astrogliosis as well as microgliosis in the ipsilateral cortex as compared to the contralateral cortex. And when looking at serine racemase expression, see that in the contralateral side, it has this more neuronal profile versus ipsilaterally, it appears to be more glial, which when we look at colocalization with GFAP and IBA1, our astrocytic and microglial cell markers, we do see this colocalization in the perilesional space. So this is suggesting that what we see in the hippocampus is likely conserved in other brain regions as well, which would be relevant to the human TBI patients that we'll be looking into. So before we get into the human data, I just want to go over how these graphs are designed. So basically on the left, we have our control samples in white, and these are tissues that were obtained from commercially available sources, usually from patients that undergo sudden cardiac arrest without CNS comorbidities. And then here we have all of our TBI patients graphed together versus here we have it separated based on region. So first we graphed all the samples from the frontal cortex and followed by all the samples from the temporal cortex. And these are just the two regions where we receive the most samples from. So we wanted to see if there are any regional differences in expression patterns. We also graphed this, color-coded this based on the time from arrival at the hospital until surgery. So in the circles, we have patients that were admitted to surgery less than a day after arriving at the hospital, versus in squares, these are patients that underwent surgery over a day after admission. And this is just to see if there are any patterns according to the time of injury. And finally, this is color-coded based on severity according to the Glasgow Coma Scale, where in red represents patients that suffered a severe TBI versus a moderate TBI in blue and a mild TBI in yellow. So when we looked at serine racemase protein expression using Western blot, we didn't see many differences in the TBI patients as compared to controls. We saw upregulation in 3 acute severely injured patients. However, overall, there were no changes. But since proteins are highly susceptible to ubiquitination and degradation after injury, we wanted to look at this from a different perspective as well. So we performed qPCR from the mRNA from these tissues. And here we do see a significant increase in serine racemase mRNA in all of our TBI patients, suggesting that serine racemase is upregulated after human TBI. We also wanted to look at if, in addition to synthesis of D-Serine, if release of D-Serine is affected in these human TBI samples. So we looked at expression of SLC1A4, which is one of the primary glial transporters of D-Serine. And here we saw a significant upregulation of SLC1A4 protein in our TBI cases. And we also did see upregulation in some of the mRNA as well. So together with the upregulation of serine racemase mRNA and SLC1A4 upregulation, this suggests that D-Serine is being synthesized and released at higher levels after injury. Then I also looked into the expression of proteins that are involved in synaptic pruning. So for example, I looked at C3 expression, which is implicated in phagocytosis of synapses. And here we did see a significant increase in C3 expression after injury. And what's really interesting is that protein levels of GPR56, which is this microglial receptor that has recently been implicated in synaptic pruning, is upregulated. Specifically, when we look at this based on region, we see that it's upregulated in samples obtained from the temporal cortex, as well as patients that were had severe TBIs and for the most part were acutely injured. So this suggests that there is this upregulation of synaptic pruning after injury, and that the regulation of GPR56 may be dependent on region and severity as well. Okay, so to summarize everything that I presented today, first, you see that the ablation of microglial serine racemase is sufficient to preserve synapses in the hippocampus, as well as rescue functional outcomes on a fear conditioning paradigm. Also, I've seen that pharmacological inhibition of the gluin2B NMDA subunit can preserve dendritic spine morphology at 3 days after injury. And complement C1q, which is implicated in synaptic pruning, is upregulated after injury in control mice, but not in our serine racemase knockout mice, suggesting that D-Serine is controlling C1q expression. And finally, looking at key components of D-Serine mediated pathology in human TBI tissues, we see that there is an upregulation as compared to control tissues. So some of the future directions that I haven't been over yet, we'll be looking at our human samples using a genomic approach, where we will perform RNA sequencing of the samples to look at acute responses to injury on a more global level. And we've also begun to obtain cerebral spinal fluid from these patients as well, where we'll be evaluating D-Serine levels after injury and seeing if there are correlates with severity of injury and outcomes to D-Serine levels. And finally, I'll be looking at the regional distribution of serine racemase expression to see if this mechanism is implicated in other areas of the brain as well. So with that, I'd like to thank everyone in my lab, especially Dan, my mentor, and Stephen, who is a previous grad student in the lab, Madeline, a postdoc who's been very helpful, especially with the human patient samples. And then a huge thank you to Jose, Maria, and Magda for all their animal and technical support. And thank you to all of our collaborators, especially Dr. Gracioli, who has been providing us with the majority of our human samples. And thank you all for listening. I'll be happy to take any questions. Okay, well, I guess if there are no questions, thank you all so much for attending. So there's a question in the chat, if I plan to look at neuroprotective effects of estrogen after TBI. Yeah, I think that's definitely something I'm interested in looking at in the future. First, I'll be kind of evaluating just overall, serine racemase expression, as well as overall D-Serine levels in the female mice. And if I do find decreases in expression, I would be really interested to see if in overoptimized female mice, if there is an increase in serine racemase and D-Serine. So seeing, if estrogen is maybe what is upstream of the serine racemase expression and kind of contributes to lower levels in female glial cells.